So before we go to the study session, Troy, let's briefly, I think this we may not be more favorable to us because it seems we are taking a lot of time. And these are things we can actually deliver within a short period of time. So I don't know, maybe we may either have to change the way we do it so that we can manage our time, how we have to change a platform if we need to go or continue the way we are doing, because the way we are doing is actually taking a lot of our time. So before we go to the study session 3, let me briefly talk about this. For those of you that are already in practice, they must have known they must have known about blood grouping system, where some organisms are said to be blood group. Well, some organisms, some individuals are said to be blood group A, blood group B, blood group O, and the combination of blood group E and B. What does that mean exactly? How could an individual have a blood group O while another person having a blood group E? Anybody with any idea?
Please, sir, please, what was your question once again?
Sorry?
Please, what was your question once again?
Why do we have different blood groups? Why an individual have O while another person having A while another person having B?
Okay, sir, I'm not sure. I just want to try.
Hello, sir.
Yes, go ahead.
Okay, I
Okay.
think it's because we have different shape of red blood cells.
Chef, Chef, in which sense? Because we know red blood cells are biochemical in shape.
Okay.
Can I try?
Yes,
All
go
right,
ahead.
can I
Go
try,
ahead,
sir?
I think it's because of the different antigens and antibodies in the blood.
Yes, let's go with the antigen. That's correct, but we explained that. Any other opinion?
Okay, so yes, we have different blood group because of the different antigen and that was what I'm supposed to say earlier. But just like every other cell information that is passed down in different families, we always have different blood groups and all in different people.
Okay, that is all correct. We talk about antigen. We are talking about antigen. The question is, what is this antigen? So if we may look at it in this way, then we just talk about the glycoproteins. And we may say these glycoproteins can be regarded here as antigen, as you were calling it antigen, yes. There are different antigen presence. How could this antigen differ from the other antigen? Now let's look at for individuals with blood group O. You have a protein. Attached to that protein on your membrane, plasma membrane, there are some monosaccharides, some sugar molecules attached to it. So we can see from the key here, after this red, there is this cycle, which is what? Galactose, right? You can see it here, galactose.
Yes, sir.
Yes. Then the next one is this blue square, which is what? Can somebody read it?
Acetylgalactosamine. Okay,
N-acetyl.
blue square. Okay, N-acetylglucosamine.
Yes, this is N-acetylglucosamine. And then we have this blue again, which we said is galactose, right?
Yes,
And
sir.
the last one is glucose. All these ones, for example, galactose is a sugar. We'll discuss it in our not next, because next class is nucleic acid. The one after it, I think, is carbohydrate chemistry. We'll know what is glucose, what is galactose, and all these, they are sugars. They are sugars. We know glucose now. Well, at least glucose we know it, but galactose and the rest, we'll definitely talk about it. Okay?
Yes, sir.
Yes. So N-acetylglucosamine is a glucose, but a derivative of glucose. What happened to glucose to become N-acetylglucosamine? We'll discuss that when we come to that. Also, N-acetylgalactosamine. Glucose is also a derivative of another monosaccharide or another sugar, which we'll definitely talk about when the time comes. So what happened here is we have this red, which is the red blood cell called erythrocyte from the key here. So attached to the protein on the red blood cell, we have sugars. The first one, we have galactose. The second one, we have N-acetylglucosamine. The third one, we have galactose again. And the last one, we have glucose. So individuals in which on their red blood cells contain this set of sugars only, they are classified as group O. So their blood group is O. But there are some individuals that on their red blood cells, apart from this set of monosaccharide found on the red blood cells, in addition, on galactose, we have some that have N-acetylgalactosamine attached to the galactose here. In O, they don't have anything. But in blood group E, they have on their galactose, they have another sugar called N-acetylgalactosamine. So those individuals are considered as group E. Are we together?
Yes, sir. We can hear you, sir.
Yes, I will agree.
Good. So the last group on their red blood cells, apart from the normal sugar as in O, they have on the galactose, instead of N-acetylgalactosamine as in the case of A, they have another galactose attached to galactose. So they are classified as individuals with blood group B. Whereas we also have an individuals that have a composition of A and B, where in their on galactose, they have both N-acetylgalactosamine and galactose.
Okay.
They have both the two. So they are A and B. So they have what is in A, they have what is in B. Therefore, they are classified as A and B. Is that clear?
Yes, sir.
So from here, we can easily see why we have donors, we have acceptors, and the rest. At least because Yes, we can somebody can tell me which among this is a universal acceptor.
AB.
AB.
AB,
Why
sir.
AB have universal or is considered as universal acceptor?
According to your explanation now, it should be because they have galactose and N-acetine.
Use the word antigen now.
Okay. So it would be because they have antigens A and B present. So there's really nothing the antibodies will be fighting against.
Yes, and in addition, you know, all these antigen A and B, they have O already in it.
Yes.
Yeah, so they have antigen A, they have B, and also O is there. Right?
Yes, sir.
Yes. While which one is the universal donor?
O, blood group O.
Blood group O, simply because
They don't have any antigen, sorry, antibody on them that
yeah.
would
So
that would give the effect problem.
so that is just the classical explanation, and from here we can also know why O cannot receive from any but O, right? And why AB can receive both from O, and why can A can only receive from O and A, why B can only receive from O and B, right?
Yes, sir.
From the little explanations here. I hope everybody got this.
Yes, sir.
I don't know, some of us are not participating. Sunny, are you with us? What of Chinazo?
Yes, I'm with you.
Okay, I like it. Veronica, are you with us? Fatima.
Yes, sir. I'm here, sir.
Please be
Fatima
with
is
us.
here.
Don't just join us and
I'm here, sir. Fatima
oh,
is here, sir.
oh good, Fatima. Okay.
Yes, sir.
So let's continue. So I think this is where we have the end of study session 2, and we enter study session 3. I'm entering study session 3 just to be able to finish up with cell structure and function, because study session 3 is also cell structure and function, just a continuation. So we continue with organelles, cells organelles, the structure in cells. Remember, we talked about cytoskeletons, right? We said we have a cytoskeletons in the cells, and those cytoskeletons are responsible in supporting the shape of the cells. They are involved in transportation within the cells. It gives the strength, and also during cell division, cell movement. I think when I asked of example of unicellular organism, an example of amoeba was given to me, and we made mention of pseudopodia, right? Somebody, I think I mentioned pseudopodia. Class, are we together?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Yeah. So these cytoskeletons, they are just structures. Example, microtubules, filaments, actin filaments, intermediate. So either microtubules, either microfilaments, or intermediate filaments. They are found inside the cytoplasm. What do? They help in maintaining the shape of the cell. After maintaining the shape of the cell, another thing they do, they are involved in the movement. For example, person that mentioned pseudopodia. In amoeba, we know amoeba does not have a leg. How does amoeba move? Amoeba form a false leg called pseudopodium. How does it form the false leg? It's due to the contractile ability of these cytoskeletons. So these cytoskeletons have the ability to undergo contraction and relaxation, thereby being able to push some part of the membrane, creating a pulse leg, something like a leg, because we just mentioned that the plasma membrane is fluidy in nature. It's very flexible. So once there is a force in one direction, it will create a kind of a protrusion that look like a leg. So that is how amoeba is able to move. So also these microtubules, filaments, and intermediate filaments are involved in transporting substances even within the cell. We just talked about, we will talk about we talk about transportation within the cells. We'll see how some component in a cell move from one portion to another. What helps them in that movement is with the help of these cytoskeletons, which we will do that. So before we talk about that, we talk about ribosomes. And I think when I mention nucleus, we talk about where ribosomes are synthesized. What are these ribosomes? Are nothing but what? Ribonuclear proteins that are produced or synthesized inside the nucleus. What do they do? They are involved in the synthesis of proteins. So these ribosomes, we'll talk about some other component of ribosomes or some other components that are involved in the synthesis of protein when we come to, if there is a need, I think in the curriculum, then we talked about the protein synthesis. But ribosomes are involved in the synthesis of proteins. Mostly they are found associated to endoplasmic reticulum. And we said, what are these endoplasmic reticulum? Endoplasmic reticulum form a kind of extension of the outer nuclear membrane. So the outer nuclear membrane hold upon itself to form what we call endoplasmic reticulum. We can see from the structure here, we have the endoplasmic reticulum, and attached to the endoplasmic reticulum, some dots, which represent the ribosomes. So the ribosomes are attached to the rough endoplasmic reticulum, which serve as a house for the synthesis of proteins. Are we together?
Yes, sir.
Yes, sir.
So we also have a smooth endoplasmic reticulum. So endoplasmic reticulum that does not contain these ribosomes. So basically, the smooth endoplasmic reticulums are the site for xenobiotic metabolism, the metabolism of foreign substances that are not known by the body. For example, when you take a drug, the drug is not known by the body. So it will be taken there into the smooth endoplasmic reticulum, and the body will process or metabolize it. Like in calcium release during contraction and relaxation of muscles. Contraction and relaxation of muscles. We also have a synthesis of lipid occurring in smooth endoplasmic reticulum. We also have lysosome. We know what lysosome is, right? A lysosome is a body that contains hydrolytic enzymes that are involved in digestion of substances. We just talked about the role of macrophages. Somebody, I think, mentioned, somebody mentioned the role of phagocytes, right? The white blood cells. After the death of red blood cells, what happened to the red blood cells? That they are phagocytosized by a group of white blood cells like natural killer cells, macrophages, etc. When they take up these substances, they are not needed in the body. How does the body get rid of it? Because they are no longer needed. Or another example, when a virus or bacteria get into the cell, the body needs to destroy that bacteria. How does the bacteria destroy it? So the bacteria is taken into the lysosome, and the lysosome contains digestive enzymes that break down the bacteria to the smaller components. And remember, while discussing in our introduction, we talked about organization of these molecules, how human being himself is made up of these elements, how these elements come together to get molecules, how the molecules come together to form these, what we call the biomolecules, the macromolecules, how the macromolecules form the supramolecules come together to form the cell themselves and the system, the tissue, the organ, and the human being eventually. So these digestive organs, the lysosome, the organelles, contain the hydrolytic enzymes. The enzymes that break down protein, the enzymes that break down carbohydrate, the enzymes that break down lipid and the rest. All of them are there. So once bacteria is taken, because the bacteria is made of all these proteins, carbohydrates, lipid, it is then broken down into the smaller fragments. How does that happen? For example, when you have this example here, it gave a cell that is no longer an organelle that is no longer an organelle that is no longer needed in the body. How does the body break it down? For example, here we can see, can we see? Are we seeing?
Yes, sir.
So we have a one out organelle here. It will come here and fuse with lysosome to form what we call a secondary lysosome. So you have the content of this organelle be released to them. Because what exactly happened is that this is a membrane. This is also a membrane. So the membrane will create a kind of pinch here and another pinch here. So they will just seal up. It will only form one membrane. So the two membranes will become one. So that the content will now mix together. Remember, the content of this lysosome is hydrolytic enzymes. Therefore, the hydrolytic enzyme will now mix with the content of the other organelle. So what is there inside will be broken down completely, crushed. You can see the broken down organelle. And here, when it is broken down to proteins, amino acid, sugars, nucleic acids, the ones that are useful, the ones that can be used, they can release out into the cytoplasm. Therefore, they can be used to produce a new organelle. But the ones that cannot be used will now, this lysosome will now push them into another lipid vesicles where they will come and join with membrane, plasma membrane, so that they can release the content out to the extracellular fluid. And this process of fusing of this membrane with the plasma membrane, thereby releasing the content, is what we call exocytosis. We'll talk about the exocytosis shortly. This is similar to in the case of bacteria, where you have a bacteria. Once the bacteria come, the bacteria will form, the membrane will cut the bacteria so that you have a vesicle containing the bacteria. So this vesicle can now move and then join with or fuse with lysosome so that the bacteria will be exposed inside with this hydrolytic enzyme. So the hydrolytic enzyme will break down the bacteria to the smaller components. The components that are needed will be released into the cell. Those that are not needed will go and fuse with the plasma membrane of the cell and then release the content out of the cell. We also have mitochondria, and we know what
Yeah, but I
mitochondria
didn't
is.
understand that.
This one.
The process, yes, the process of phagocytosis.
You have, for example, when you have a macrophage, we know what is macrophage, right? The white blood
Yes.
cells that is involved in taking off foreign substance and break it down. So inside the macrophage, there are lysosomes, the organ we are talking about. And we say this lysosome is like an incinerator where unwanted substances are burned, broken down, destroyed. Clear? Is that clear? So
Yes, sir.
when you have a bacteria now, and bacteria coming into the cell will recognize it as maybe using the antigens, all these glycoproteins will now recognize that this is a foreign substance. This is bacteria. This is not needed. This is not welcome. So how do we get rid of it? Then we form a membrane. The membrane will engulf it. It will form a small membrane inside into the cell. This process is what we call endocytosis. A new substance is coming, and they're recognized by glycoproteins. And now glycoproteins form a kind of a sachet for it, a sachet of membrane. So a membrane is formed around it, so that yes, it is taken inside, but it is enclosed within a membrane. And this is possible due to even the flexibility we are talking about of the membrane. If the membrane is not flexible, this is not possible. But because membrane is flexible, it can easily fold over itself and pinch out of the main membrane so that it can seal on its own using the what we call the self-healing process, so that it will form a small vesicle inside. And this vesicle, lipid vesicle containing the bacteria. Are we together? So
Yes, sir.
inside now the foreign substance is already known that no, this lipid vesicle contain bacteria, something we don't know, so we don't want. So how do we destroy it? So it will come and fuse with lysosome. So lysosome also contain membrane. So it will just, the membrane of the lysosome will create a pinch, and also that of the vesicle will create a pinch, and they will become one. They will join up together. They will become one, so that the component of the lysosome and the component in the vesicle will now become one, as we can see here. The two are coming together and they are becoming one. So after they have become one, we can see this bacteria, because the hydrolytic enzymes come in contact with the bacteria, it is broken down to pieces. Is that clear?
Yes,
Once
sir.
it is broken down, meaning it is destroyed to pieces, then the ones that are needed by the body, maybe the proteins, the amino acid, the sugars, the nucleic acid, can then be released inside the cytosols and then used by the cytosols. The ones that are not needed will now come down closer to the cell membrane again and create a pinch. After creating a pinch, then the cell membrane will heal, and then this content will get released out of the cell in the process we call exocytosis, just as here in this case. Are we seeing?
Yeah.
So this is just the explanation I gave. In mitochondria, we all know what mitochondria is, right? The powerhouse of the cell where energy is produced. Yeah, we're also running out of time, so let's see how we can rush before we listen. So this is where energy is produced. The mitochondria is also a double membrane. It contains an outer membrane and the inner membrane. The outer membrane allows substance to go in and out, while the inner membrane doesn't allow substance to go in and out, because the inner membrane contains some proteins, and those proteins are involved in the synthesis of energy. I think if the curriculum allows, we also talked about how energy is synthesized in the mitochondria. So the next one is Golgi bodies. The Golgi bodies are just like vesicles and sacs that are involved in modification. One, for example, protein and lipids are synthesized from endoplasmic rough and the smooth endoplasmic reticulum. they can be transported in form of lipid vesicles to the Golgi bodies. We can see that it has the two spaces, the trans and the C space, where in the C space, you have an incoming vesicles coming in containing the newly produced protein or the newly produced lipid, where they can be modified, where they can be packaged so that they can perform their own function. After passing through the cisternate of the Golgi bodies, then they will be packaged, they will be modified, and then ready to be taken to where they will function. Then a newly formed vesicle will be formed containing these proteins and the lipid, where they'll be transported to the site of action. So you can see the role of these cytoskeletons in the movement of all these vesicles from the where they are synthesized from the endoplasmic reticulum to the Golgi bodies, from the Golgi bodies to the point where they function. What to move them? What to move them is the cytoskeletons. Well, the last thing we'll talk about is the cell membrane, which we've already talked about, and I think what remains is the transport across cell membrane. Remember, we mentioned one of the functions of the cell membrane is the transportation, right? The transportation of substance in and out of the cell. It regulates the transportation of substance in and out of the cell. Is that clear? One of the major functions, apart from the protection, apart from the separation between the intra and extracellular fluid. Now, how does the membrane facilitate the transportation of substance in and out? We say there are three methods. There is passive, there is active, there is bulk. In passive, simply the transportation of substance in or out of the cell membrane that does not involve energy. Rather, it only involves a diffusion process. And we say this diffusion process is classified into two. Could be simple diffusion or facilitated diffusion. It is regarded as simple diffusion if the substance that goes in or out of the cell is very small hydrophobic or hydrophilic substance that can easily dissolve in the membrane and then move in or out of the cell membrane. We give an example of CO2, alcohol, water, oxygen, water, etc. They can easily, because they are very small in structure, they can easily dissolve in the membrane and then pass through without, by just a simple diffusion process. Once there is high concentration in one side, they can easily diffuse into. And we know what is diffusion, all right? Diffusion is a concentration-dependent phenomenon. Is that clear? Is that clear?
Yes, sir.
Yes. So in facilitated diffusion, usually is also a diffusion, but involve a carrier, where a carrier protein is involved in movement of this substance in or out of the cell. This is just the difference. They are also controlled by diffusion processes, concentration gradient. Once the concentration of substance is high outside the cells, there is possibility that they can easily move in. But this movement with the help of a carrier protein. We talk about integral proteins, and these integral proteins can serve as transporters. So basically, they are the ones that are involved in movement of these substances. Sometimes it can be a uniport, where you have, for example, substance like glucose or amino acid can move in through a particular transporter protein through diffusion due to concentration gradient. In some cases, you can have what we call an antiport. An antiport where movement of substance facilitates the movement of another substance. Using the same carrier protein, something is coming in, something will go out at the same time. That type of transportation is called an antiport. One is coming in, will facilitate another thing to go out. One molecule is coming in, another molecule is being transported out by the same protein. It's antiport. It's import when you have the two molecules. One is coming in, because that one is coming in, the coming in of one will facilitate another, so that two of them will come in. Another one wouldn't have come, but because of this one that is coming in, it will facilitate another one so that two of them will come in. We have what we call a syncope. Are we clear? Are we clear?
Yes, sir. Yes,
So
sir.
just trying to
Yes,
give you a
sir.
summary so that it will not take much of our time. We have less than 3 minutes for the session also to close. I don't want to also create another link and start it all together. So I'm trying to round up this so that even if it is closing, we can use the WhatsApp chat to round up and then answer questions. So inactive is different from passive in the sense that inactive energy is involved. For the movement, for the substance to move in or out of the membrane, it requires energy. Why does it require energy? It is because in most cases, the substance that need to be transported is big in size. Or the movement is against concentration gradient. Maybe there is high glucose inside and another glucose that is outside want to come in. So he want to go against the concentration gradient. So it's very difficult for him unless if an energy is used. So that energy will push him inside. Is that clear? So it can be primary, it can be secondary. In primary is when the energy molecule called ATP is used. While in secondary is when a concentration gradient is used. When the concentration gradient is used, maybe the high concentration of, there is high concentration of potassium inside the cell and the sodium is coming in, then potassium will go out and then sodium will go in, and in that process, an energy is released. That energy is used to facilitate the movement of another substance inside the cell. Is that clear? That is secondary active transport. And the last one, we call it bulk transport, the transport of substance in bulk, many molecules being transported inside the cells. And we have endocytosis, we have exocytosis. I've already, what is endo and exocytosis? Endocytosis is when something is coming in, bringing into the cell. It could be phagocytosis, which just we explained. It could be pinocytosis, which involve the movement of liquid into the cell, or it could be receptor-mediated. I think we explained receptor-mediated while trying to explain liposomes in liposome-targeted therapy, for example, in cancer, where we say it is the receptor on the surface of the membrane that will mediate the, the intake of the substance inside the cell. And we have exocytosis, where substance get out of the cell. So we can see phagocytosis also, which I already explained here, and pinocytosis. And here we have receptor-mediated, where it is the receptor that facilitates the movement of the substance in and out of the cell. And this is classical exocytosis, where we have the vesicle coming together and create the pinch, and then the substance will get released out of the cell. So the next, I don't know, we will not have a time to ask question, but I will shortly give a video call through WhatsApp, so that because I have less than 1 minute to finish up with this, so that we can have a questions and answers there,